In this video, I am going to do a quick demonstration of how you can use the Reactome pathway database to look up different pathways that may be affected by the supplement of your choice of research, and how you can look at different pathways links to each other or their interaction with each other. So the first thing you need to do is have your list of proteins and genes ready that you found through your literature research in order to look for the pathways associated with them. You would go into the Analyze Data area of the Reactome tool. When you go to the Analysis tool, you will see that it asks you to put in the data that you want to use to analyze. Now, this can be in any different form or a bunch of different forms. The ones that we are going to be using is pretty easy one. We're just using the UniProt accession numbers, which are unique protein IDs that are given to each protein. And we will be looking those up using the OMIM database from NCBI. I will show you how to do that in just a second. This is going to give you, enough information for now for this type of analysis, just to run a simple analysis. However, this program or this platform can be used to run much more in-depth and much more involved analyses, especially when we have microarray data or gene expression data from various time points or multiple treatments. It can actually be used to look at differential gene expression and to look for a lot of cool things. So that's for later though. We are just going to run a simple analysis. So to look up UniProt or the unique protein IDs, we will go to this NCBI website. So let's say that I found P53 as one of the genes expression that is changed in response to my treatment. I can look it up in this OMIM site of the NCBI, and then I want to make sure that I look up the human one. I'm going to open the TP53. Here it gives me the general information for it. On this right side, you're going to see external links, and it's going to give you a lot of different options. It has genome, DNA, protein, gene info. So you're going to go to the protein area and you will click on UniProt. When you do that, it is going to open up a new link, and that is going to contain the UniProt ID that you need to use. So I'm going to copy this and I'm going to be using that. So I'm not going to use their example. I'm just going to be putting in my own. And you're going to be making a list of these. So if you have 5 different genes or proteins, you'll put 5 different ID numbers in there, one on top of the other. I'm going to just do an example with a couple of them. So I'm just going to now once you're in here, you can just go ahead. I think you should be able to go ahead and put all your others directly in this area. And when I put BAX, it gives me all different kinds of BAX genes. I want to click on the human one. And here's the UniProt ID for it. I don't even have to click it. It shows up on that as well. So that's going to be my second one. And let's say my third one was, I don't know, I'll come up with another one. Let's do P21. So we'll do P21. Search for that one. Again, it's going to open up a lot of different ones. I want to pick, no, this is not the right one. P21 protein. Okay, we'll pick this one. And we'll go to protein, UniProt, because I got out of that site, and make sure it's human. And now I can select for this one again, and I can put it in my Reactome. So I'm just going to go ahead and show you the example with just 3 of these. I will click continue, and then you want to make sure to click on this box that says project to humans, so that it can give you data related with human genome, and say analyze. Now, the first thing you're going to notice is this area. This is showing all the networks that are changing in response to these 3 proteins. And with these 3 proteins specifically, you see changes in immune system, DNA repair, cell cycle, programmed cell death. Obviously, all of those are important pathways that we know P53 is responsible for. And that happens because it is a transcription factor, which is why the gene expression or transcription pathway lights up. And the signal transduction pathway lights up because it does the work that it does through signal transduction. So it is, again, like I said, extremely important protein. Now, if you look underneath, you will notice that it's going to give you all the pathway names that are associated with these 3 proteins. And you will see that it will also have this thing called entities found. So it says 2 out of the 3 proteins were somehow involved in this particular category. When you click on this, it's going to automatically light up the appropriate areas on this network chart to talk about where it is that you can see those. You also had the p-values in there associated with that as well. I actually clicked out of it, unfortunately, so I'll fix that. Okay, so once you know all the networks that are changing based on the proteins that you put in, the ones that those proteins that you put in are associated with, you should focus on the ones, gene expression and signal transduction are disease. These are just general networks. So they're too general, too macro. Don't worry about them. Look at these more specific networks and choose the 3 most important ones that appear to come up. In this case, the DNA repair, cell cycle, programmed cell death might be good to look at. So for any one that you want to focus on more, you would click on that particular network and look for details of that particular network. And it will now load a diagram that is more in depth in that particular network and show all the pathways associated with it. And if you look down below here, those will pop up in here as well as the apoptosis and programmed cell death, intrinsic pathway of apoptosis. So you know exactly where those networks, which pathways are exactly involved in that particular network. It takes a little bit of time sometimes for the diagrams to load, so please be patient. Okay, so one of the things that you can notice, it lights up the apoptosis area over here. It also lights up the appropriate areas on this left-hand side in the event hierarchy. If you look in here, you can actually find more details about that in here as well. So right here, you can see that 2 out of 2 different proteins associated with apoptosis are in your data set out of a possible of 180 proteins that are involved with apoptosis. All the proteins that are in your data set involve intrinsic pathway of apoptosis. So this tells you that your drug or supplement works through intrinsic pathway for apoptosis. You can click on this, and this actually works a lot faster than going into the program itself and clicking on them, so that it will give you more details about this particular cascade, and it will load a diagram associated with it, and it will show you how it works. You are looking for areas that have this yellow, one of these colors light up to show how significant it is in your data set, and that's intrinsic pathway. So that's what you want to focus on. And when you open it up, you see exactly now, you see all the different areas that light up that will be associated with either P53 or BATs. So you can use that to figure out what exactly you will be including in your final schematic that you will prepare yourself. Once you're at that level, you can download these by using export diagram tool, and you can export it by saying as a diagram, and it gives you a little bit more macro view, or get a snapshot which gives you this diagram where you can actually see where each section is. So this is actually a more useful diagram, so you can download that as a PNG to use for yourself, so you can refer back to it as you're building your own schematic. Now if we go back out a little bit so that we can see it a little bit more macro, well, I went back in here. I can go further into it by clicking on different sections individually and look at it more in detail. Now again, I just went a little macro into apoptosis, so obviously it's showing me more general observations. And I want to see the intrinsic pathway more. There is a place where I can click on to get more details from it. If I don't want to look at the one before. Otherwise, if you were to look at just this portion, that's the portion that you care about, because that is the intrinsic pathway. You can also, if you have trouble, because sometimes it does give trouble going into the exact pathway, you can just look that up separately as well once you identify the exact portion of the pathway that you care about. Now, when you make your own schematic, you can essentially copy the same idea and make it in the PowerPoint using the same type of arrows and shapes, or you can make your own, build your own as you go. In your final schematic, you want to make sure that all three major parts of your pathway and the networks that were associated, all three major proteins that you put in, and the pathways and networks associated with them are represented. And so there will be a separate video where I will describe how you make those PowerPoint slides, where you will combine those together. But at the end of the day, you want to ensure that you have all your initial proteins represented in the final PowerPoint slide schematic, along with the pathways and networks associated with it that you find through the Reactome database. Okay, hope that helps.